Hello, in today's video we'll be exploring ultraviolet light. The UV spectrum refers to the portion of the electromagnetic spectrum that's short of invisible light but longer than X-rays in wavelength, ranging from 10 to 400 nanometers in length. As we will show in these experiments, it can cause fluorescence in certain materials, which has multiple uses in science. In today's experiment, we will need 80 milliliters of water, a strong purple dye called eosin, sodium hydrogen sulfate, and sodium carbonate. And because we'll be dealing with fluorescence, we'll need to do this experiment in dim light. Firstly, we'll add 3 drops of eosin into water and mix, resulting in a brown solution. Eosin is a strong dye, often used as a stain in histology, its negativity allowing it to bind to positive components in the cell, such as positive proteins in the cytoplasm and muscle fibers, staining them in shades of color from pink to red. Shine UV light on this solution, it will glow yellow-green. This is because of eosin's pi bonds, which allow electrons to be delocalized in the molecule. This makes the electrons easily excited by UV light, absorbing its energy and then releasing it as visible light when going back to its lower energy states. After this, we added NaHSO4, or sodium hydrogen sulfate, which disrupts the delocalized electron system by protonating the COO- carboxyl group into COOH, which cannot share electrons into the system as well as it could before. This is because the hydrogen bond in COOH keeps electrons localized in one place, while in COO-, the lone pairs on oxygen are able to share electrons within the system. As a result of this disruption, when we shine UV light on the solution, now the glow is gone. Then we add NaCO3, or sodium carbonate, which deprotonates the COOH back to COO-. This is because NaCO3 is a base, while eosin, with a protonated COO- group, is an acid. As a result of this change, the yellow glow returns as the delocalized electron system is no longer disrupted. In this next experiment, we must first crush some leaves using a pestle and mortar to break down cell membranes and cell walls to release chlorophyll. We then add the result of this crushing into a vial. and then we add triethyl citrate in order to separate the chlorophyll out. This is possible because both chlorophyll and triethyl citrate are soluble lipids and will therefore interact, drawing the chlorophyll out into the liquid. This interaction is slow, however, so we need to use a hot water bath to speed it up by increasing the temperature for 10 minutes. After taking the vial out and shining UV light on it, you can see that there is now a red glow. This is a result of UV light exciting electrons in chlorophyll, which, just like eosin, releases energy in the form of visible light when it goes back to its ground state. Fluorescence, like what we've discussed in this video, can be used for purposes such as analysis of proteins and staining cells, as I explained with eosin. Furthermore, UV light can also be used to cause chemical changes in compounds, making it useful for even more things from sterilization to causing damage to cells that allows researchers to study their DNA repair mechanisms. Thanks for watching.